Hi, and thank you so much for the invitation to be here. I'm really excited for all the speakers in this workshop. So my talk today is called Idiosynchrony, From Shared Responses to Individual Differences During Naturalistic Stimulation. And so to motivate this line of work more generally, again, hopefully, if you're here, you are already interested in this topic, but I think the idea of using naturalistic stimuli in neuroimaging to study individual differences is a really, really interesting one. And so there's something that's known in the social sciences as the Rashomon effect, and it's named after a 1950 Japanese film in which there's basically a pretty gruesome crime, and there are four quote unquote eyewitnesses to the crime, but they all have very different accounts of what happened and very different interpretations. And so this is a phenomenon that's pretty easily recognizable in our daily lives. When faced with the same sensory input, different people will arrive at different conclusions or different reactions. And that's particularly true when the information is sort of complex, high level, ambiguous in some way. Although it also happens with low level perceptual phenomena. And so I think one of the really great opportunities with naturalistic stimuli is that they allow us to tap into some of these processes that are most likely to vary across people. They allow us to present people with precisely the type of information that's most likely to evoke different reactions in different people. And then we can then study the process of online impression formation in the brain as it's occurring using neuroimaging. And this is an interesting thing to do, I think, not only from a basic psychology, basic cognitive neuroscience standpoint, but also from the standpoint of understanding mental illness. So mental illnesses, various mental illnesses have been associated with different cognitive biases in terms of the light in which people tend to interpret things or assign valence to things, for example. And so naturalistic stimuli represent an opportunity to start to understand those tendencies and those biases, not only in the normative population, but also in people with or at risk for mental illness. So with that motivation in mind, I'll give you an outline of what I plan to talk about today. So the talk will be in 3 sections. In the first section, I will ask and answer this question, hopefully, of can we detect stable and meaningful individual differences during naturalistic stimulation? And I'll define what I mean by stable and meaningful. In the second section, I'll talk about this question of can we design a bespoke naturalistic stimulus to draw out individual variation of interest? And finally, I'll spend a little bit of time talking about how we can think about modeling individual variability during naturalistic stimulation. So to start out with this first section, can we detect stable and meaningful individual differences during naturalistic stimulation? And the reason this was an interesting question, at least in part, was because as you've hopefully gotten a sense for by now, these naturalistic stimuli have been used for close to 2 decades now in human neuroimaging, and usually with the goal of studying shared responses across people. So in other words, beautiful work showing that in a group of people watching the same movie, you see this really strongly synchronized activity across different brains. And so it was kind of this open question, does that saturate all of the individual differences, or are there still stable and meaningful differences that we can pick up on even during these really rich, evocative naturalistic scan conditions? And so to introduce this part of the talk, most of the analyses I'll be presenting in this first section are based on functional connectivity, which many people are probably familiar with, although there are a lot of different ways of doing this type of analysis. So just to get us on the same page, the analyses I'll show here in this first section were done using a whole brain atlas. So essentially we're dividing the cortex, subcortex, and cerebellum into a series of regions. And then for a given pair of these regions, we can look at the fluctuation in their signal over time and calculate the correlation there. And if we do this across all possible pairs of nodes in the brain, we can get this whole brain functional correlation or connectivity matrix. And we can do this for a given individual during a given scan or a given brain state. And so one result that we noticed a while ago when we were hoping to use functional connectivity to study individual differences is that these functional connectivity matrices within a subject are actually stable and unique enough to serve as a type of fingerprint. And so what this looked like in our hands when we first noticed this result, we were using data from an earlier release of the Human Connectome project. And in this data set, people came for 2 days of scanning where they had a resting run and several different cognitive tasks. And we were asking this very simple question of essentially, if we calculate a functional connectivity matrix for every individual in every scan condition or task state, can we match a correlation or a connectivity matrix from the same individual across different days and across different brain states? And the answer to that was yes, we were able to do that with actually a striking degree of accuracy. So between 54 and 94% accuracy, depending on the particular pair of scans that we used. And this was much, much higher than chance. And this was a really interesting and striking result in some ways. When we looked to see where in the brain most of this identification power was coming from, we saw, as you might predict, that the networks that were most discriminating of individuals were these ones that are highlighted here in purple and teal, that were in the association cortices. So basically networks involving the prefrontal, temporal, and parietal regions were had the most individual variability and contributed the most to this identification result. We also saw, as part of that study, that these functional connectomes were able to predict a real world output of the brain. So not only are there these stable differences, but they are actually meaningful for behavior. And so what that looked like in this first study, again, was that we had a measure of fluid intelligence that everyone in the data set completed based on this progressive matrices task. We used a pretty simple machine learning algorithm called connectome-based predictive modeling, where we're training a model on a subset of subjects' resting state connectivity matrices relating strength in various connections to their score on this test. And then we were able to see that we actually were able to predict based on, in a novel subject, based on just their resting state connectivity profile, how well they did on this test of fluid intelligence. And these were predictions based on the whole brain. Again, when we looked to see where the most predictive power was coming from, we saw that the same networks that were the most discriminating of people were also the most predictive of fluid intelligence. So these sort of frontoparietal association networks. And so this was a really interesting and encouraging result for those of us that are interested in individual differences. But it did kind of raise this question of how much does brain state matter for studying these differences? So if we can identify people regardless of what they're doing while being scanned, and we can predict this sophisticated measure of cognitive ability, this fluid intelligence measure, just based on resting state connectivity, well, then does it really matter what we have people do while they're in the scanner? To make a long story short, it does matter a lot. And so I personally became really interested in the potential to use naturalistic scan conditions to really target and draw out variability along certain axes. But before we could get to that, we thought that naturalistic scan conditions, so things like movie watching or story listening that you've already heard a lot about, we thought that these would be a really interesting challenge to our fingerprinting framework, because, as I mentioned, most of the work to date using those stimuli had been focused on how they tend to really evoke these strong shared responses. So among a group of people watching the same movie, these rich dynamic stimuli really evoke a lot of, really make people look quite similar, to put it simply. So could our functional connectivity fingerprints survive this sort of saturation with this really rich and engaging task that does tend to synchronize everybody's brains in pretty convincing ways? So lucky for us, in collaboration with Tammy Vanderwall, we were able to test this empirically. So Tammy had a data set of 34 healthy subjects that came for 2 different scan sessions. In each session, they did 3 different runs. So there was a resting state run, as well as 2 different movie stimuli. So one of them was this Inscapes, which is, if you're not familiar, it's a very cool but very sort of abstract movie, originally designed to promote compliance among children in the scanner. So it's just kind of a series of geometric shapes that move around the screen and change color and things like this. It's quite relaxing, actually. And then the other movie that Tammy used was a clip from Ocean's 11. So I'll just play this again for you. Both of these are about 7 minutes long. I'm just showing a quick clips here. So these are both movies, but they're really different. So Ocean's 11 is a Hollywood style movie, really rich, fast paced, lots of dialogue, etc. Both of these did have sound, which I'm not playing. But essentially our question was, can we identify people based on functional connectivity across rest and across these different movie conditions? And we found that we could, in fact, identify people between movie watching scans as well as from rest to movies. And so this matrix here is showing the identification rate. So how accurate we were at matching individuals based on their functional connectivity profiles in different pairs of scans. So for example, we were 91% accurate in matching the REST1 scans to the REST2 scans. We were 94% accurate in matching the Inscapes 1 session 1 to Ocean session 1. And so broadly, all of these are well above chance. So this suggests that movie watching does preserve individual identifiability. Highlighting here on the diagonal, these are pairs of scans from the same condition. So in other words, REST1 to REST2, Inscapes 1 to Inscapes 2, Oceans 1 to Oceans 2. And it's difficult to do statistics on this because we really only get one number from the whole data set. But you can see that if anything, numerically, we actually do better for matching within movies. So up to 100% accurate matching from Oceans 1 to Oceans 2. And even if you look at the off diagonal, which is showing the rates between rest, going from rest to a movie or between two different movies, these are all highly accurate. And so this was really encouraging to us because even though we know that movies sort of make everybody look more similar to one another, they seem to also be, even on top of that, preserving or potentially even enhancing the within subject similarity. So you look more similar to everyone else while you're watching a movie, but you also in some ways look even more similar to yourself. And this was an encouraging result for those of us that were hoping to use naturalistic stimuli to study individual differences. So kind of in parallel to this work with naturalistic scans, our group and other groups were also noticing that traditional tasks do better than rest when talking about behavior prediction. So one thing is the identification result, but what we were even more interested in actually was how well can we predict behavior from functional connectivity? And what we were seeing on this paper in particular comes from Abby Green et al. But essentially what they found was that just by inputting connectivity data from a task state into the model, they were able to much better predict variation in fluid intelligence. So the results that I showed a few slides ago were based on resting state connectivity, where we were able to get significant prediction accuracy. But it turns out that if we use task-based connectivity, we can do much, much better, in some cases up to 5 times the variance explained. So in these graphs here, this is, they're varying the input to the model connectivity calculated during different brain states. So for example, gambling, task, working memory, emotion, motor, language, and the 2 rest scans are out here on the right. And so what you can see is that pretty convincingly, we do much better. We explain much more variance in fluid intelligence when we're looking at connectivity during a task as compared to rest. And they saw the same results in a developmental sample, this Penn neurodevelopmental cohort, where the working memory and the emotion scans did much better than the rest scan for predicting fluid intelligence, even in the same exact subjects. So we thought this was really interesting, and it kind of raised the next logical question, which was, okay, if we can identify subjects during movie watching just as well, if not better, than we can during rest, we're seeing that these traditional tasks are doing better than rest for predicting behavior, then does movie watching also better predict behavior as compared to resting state? And so luckily, we had another publicly available data set, also from the Human Connectome projects, that we could use to answer this question. And so because this was sort of a broad exploratory study, we decided to try to predict 2 kind of summary scores of behavior in 2 different domains. So one was essentially the first principal component from all the cognition measures, which were highly collinear with one another. So sort of general cognitive ability was our first prediction target, and our second prediction target was a sort of composite measure of socioaffective traits that was calculated from the NIH emotion toolbox. So broadly, does movie watching better predict behavior compared to resting state? And is there any difference between predicting behaviors in these 2 different domains? So in this study, just to quickly orient you, again, people came on multiple days of scanning. We were most interested in the sessions that contained movie watching runs. And so each of these sessions began with a 15-minute rest run, and then there were 2 different movie scans, so a total of 4 different movie scans. And the important thing to know about these movies is that these larger movie runs were actually comprised of individual video clips that were about 3 to 4 minutes in length, and that were separated by 20 seconds of rest. So these were all unique clips sort of going into these 15-minute movie runs. And essentially what we did was we just pitted the rest data against the movie data to see how well it would predict individual differences in these two domains. All still based on functional connectivity, by the way. And so what we saw was that movie watching did, in fact, in most cases, outperform rest for predicting behavior. So these are the predictions of the cognitive ability summary score. These graphs, so on the x-axis, we have the data going into the model, so rest or different movies. Dark gray is a rest run, purple is a movie run. On the y-axis is the prediction accuracy for cognitive ability. And so we ran 100 iterations of each model just to get a sense for stability of the prediction accuracy. So that's what these dots are here. And then we compared all of that to null models created with permutation testing that are shown here in light gray. So overall, you can see that although we can get significant prediction of cognitive ability, at least from this first resting state run, we, and in both sessions, actually, the movie runs outperform the rest runs, in some cases by a significant margin. So this movie 2 really doing quite well for predicting cognitive ability. When we look at the same thing with socioaffective tendencies, we see that this score is overall harder to predict. And it's important to point out that this is a self-report measure, whereas the cognitive ability was all based on behavioral tasks. But to the extent that we can predict this measure at all, we can only do so using connectivity from this movie 1 run. This was some interesting evidence that movie watching does, in many ways, outperform rest for predicting behavior. I should mention here, not including this for reasons of time, but we did largely rule out, we think, differences in low-level arousal between the two different types of scans. So, one sort of obvious question is, well, are people just sleeping during rest and they're more likely to be awake during movies, and that could explain the advantage for movies. We did have simultaneous eye tracking data for the majority of these scans, and so we were able to kind of look at measures of blinks and microsleeps and things like that. And we didn't see strong evidence that the advantage for movies was driven by low-level arousal differences. So we're fairly confident that these differences are being driven by the way that movies sort of modulate ongoing cognitive state. And as I mentioned before, the interesting thing about these movies is that, or these larger movie runs, is that they were comprised of these individual video clips that are about 3 to 4 minutes in length. And so we wanted to know whether the content of the movie mattered for predictive accuracy. And so to do that, or to get at that, we ran a follow-up analysis where we restricted the model input to only data from individual clips at a time. And these are all matched for duration. They're only about I think 2.5 minutes each by the time we had to truncate down to the shortest clip. So the interesting thing here is that you do see actually a lot of variability in terms of how different clips are doing for predicting cognitive ability. And again, these are the same subjects and same behavioral scores, but just depending on the movie content, we're seeing in some cases really dramatic differences in how the individual clips are performing. And interestingly, 2 of the best performing clips, one of them was a clip from The Social Network, and one of them, coincidentally, similar to Tammy Vanderwell's study, even though they weren't necessarily chosen to be the same, was a clip from Ocean's 11. When we do the same thing for the emotion score, again, we see overall that this score is harder to predict. So many clips did not give us a significantly accurate prediction of this score, but to the extent that we could predict this score, actually the 2 highest performing clips were similar to the cognitive ability. And again, we see sort of this interesting variability in accuracy, suggesting that the content of the clip really does matter for drawing out certain differences. And so as I kind of alluded to when we were just looking at these results, it suggested to us that perhaps the clips that were highest in social content were doing the best for this behavior prediction accuracy. And so to kind of operationalize and test that, we extracted essentially just for each clip the number of TRs containing at least one face. So essentially what percentage of the time is there a face on the screen? And that was just a very simple proxy for the amount of social content in a clip. And it turns out that when you correlate that with a clip's prediction accuracy, you see this positive relationship such that clips that are higher in social content do the best for predicting, in this case, this is cognitive ability. And just to give you a feel for these differences, so these three clips that did the best here from Social Network, Ocean's 11, and Inception, these are all, you can see multiple faces on the screen, lots of dialogue, fast-paced, and the clips that did the worst for prediction tended to be the ones that didn't have a lot of humans, were more sort of nature documentaries, montages, etc. And so we thought this was really interesting, especially or intriguingly, even though this measure of cognitive ability wasn't necessarily explicitly social. So it's not necessarily social cognitive ability. This was a composite of things like working memory and fluid intelligence. And yet we're seeing that the social clips are really somehow bringing out these connectivity differences that are related to this cognitive ability. And so one thing we wondered was whether the advantage for the social content was being driven by sort of general attention or engagement. So if people are just more likely to be interested, more likely to be engaged, more likely to be paying attention to the clips with social content, maybe that could be driving higher accuracy without necessarily being related to the specific content on the screen. And so unfortunately, we didn't have any sort of behavioral ratings or debrief measures where subjects were asked, how interesting or engaging they found each clip. But what we did have was this data from the eye tracking, the simultaneous eye tracking as people were in the scanner and watching these movies. And so we reasoned that it could be the case that if this were due to kind of a general attention mechanism such that the clips that were more engaging gave us better predictions, that maybe a proxy for engagement could be how similar people were in their eye gaze patterns. So in other words, if everyone's kind of tending to look at a similar place on the screen as they watch these movies, that could be a proxy for how engaging they find the clips, and maybe stronger synchrony across people in gaze would be an indicator of a better prediction. And so to test that hypothesis, we calculated this eye tracking inter-subject correlation. So essentially over time, where is people's gaze going? And we can do that basically. So we did have some missing data, as you can see, but to the extent that we were able to get clean data, we were able to do this across all pairs of subjects. And then you basically get this subject by subject matrix of how strongly synchronized people are in their eye movements while they watch a clip. And interestingly, we did not see, so our hypothesis again was that we might see stronger synchrony in gaze in the clips that did better for predicting cognitive ability. We didn't actually see a relationship between the median eye tracking ISC and the prediction accuracy, but we did see a relationship between gaze synchrony and prediction. So in other words, the clips where some pairs of subjects were strongly synchronized in their eye movements and other pairs of subjects were less synchronized, those were the ones that did the best for predicting cognitive ability. And so this is pointing at not necessarily a general attention or engagement. So it's not necessarily that everybody is more engaged, but it's that pairs of subjects are more engaged in similar ways. So different people, I guess, are engaged differentially if we take this eye gaze proxy as a sort of indirect measure for how engaged people are or in what ways they're engaged. And so, we'd like to follow up on this with more targeted hypothesis-driven work, but this is an interesting suggestion that it could be the case that the clips where people are paying attention to different things or people are differentially engaged, those are the clips that are really drawing out the most variability in functional connectivity as related to this cognition score. So to summarize this first section, which is also the longest section, by the way, can we detect stable individual differences during naturalistic simulation? I'm going to say yes, in the sense that identifiability or fingerprinting is preserved during movie watching. And also that when it comes to functional connectivity-based behavior prediction, we're seeing that movie watching outperforms rest for that purpose. And so sort of another question we wanted to ask with this, sort of in a more prospective design, was can we come up with a bespoke naturalistic stimulus to draw out variation along a specific phenotypic axis that we care about? And so the first trait that we decided to go after was trait paranoia. So paranoia is an interesting one. We know that in its extreme, it's associated with schizophrenia and other psychotic illnesses, but it also exists on the spectrum, even in a normative population. And so in this study, what we did was we came up with an original narrative that was designed to be deliberately ambiguous. So it was supposed to sort of stratify people according to their interpretation of this stimulus, such that people with higher baseline trait paranoia would get a more suspicious or paranoid read of this, whereas people with lower paranoia might get a more neutral or even positive impression. And to give you a very brief synopsis of the story, it was about an American physician who is sort of drawn down to be a doctor in this rural village in the Peruvian Amazon. And she goes there under mysterious circumstances. There's sort of a series of events that occur that sometimes make it feel like she was lured there under false pretenses. Other times, the events are, again, more neutral or even positive. And then the story kind of leaves off at a cliffhanger, again, with the idea that some people will get a very suspicious read of this, other people's not so much, and that's related to trait paranoia. So subject in the study came for 2 visits. The first one was purely behavioral, where we got several different trait measures, including trait paranoia. They came back about a week later for the fMRI visit. And so again, everybody heard the exact same story. This was an audio recording of the story that we had written. So everyone's getting the exact same sensory input. And the idea is that people are arriving at different interpretations of that input. And so moving away a little bit from functional connectivity, in this study, our main data analysis approach was intersubject correlation, which hopefully you're familiar with by now. But again, just to make sure we're on the same page. So this intersubject correlation, or ISC, is this very elegant technique where essentially if 2 people are exposed to the same naturalistic stimulus, they're getting the same sensory input over time. If you see a correlation in neural activity in the same region across 2 different people's brains as they're experiencing the stimulus, you can infer that that region is somehow responding reliably to the stimulus. And in this study, we extended this framework to ask not just which regions are responding reliably in all subjects, but rather which regions might be responding more reliably in certain groups of subjects, and specifically in subjects that had higher trait paranoia. So before I show you the paranoia results, I'll just show you ISC across the whole sample. So we saw replicating past work by many other labs with these types of stimuli. We saw that not only did we see high synchrony in primary auditory cortex, but we also saw strong ISC in regions of association cortex in temporal, parietal, and prefrontal areas. And one of my favorite things about this map is that outside of A1, some of the peak locations of synchrony, so there's one here in the sort of posterior superior temporal sulcus, or the TPJ. And if you do a neurosynth or a meta-analysis search for the top terms associated with this voxel, you get theory of mind and mentalizing. And same thing for this region down here in the cerebellum. So this is sort of some kind of a hint that as subjects are listening to this story, they're potentially engaging in the types of cognitive processes that we were hoping that they would. So thinking about characters' mental states, trying to attribute intentions and make sense of these social interactions. But again, we were most interested not necessarily in the regions that were synchronized in everybody, but we were more interested in the regions that were synchronized more so in high versus low paranoia participants. So to look at this, we stratified our sample into people with high trait paranoia and people with low trait paranoia. And then we looked at regions where pairs of high paranoia participants were more synchronized. And we did this at a whole brain level, and we found 4 regions that emerged as being more synchronized in the high paranoia group. And I'm showing you here the name of the region, as well as the top neurosynth terms associated with that region. So we saw stronger synchrony for highly paranoid people in the left temporal pole, the right medial PFC, the right dorsomedial PFC, and the left precuneus. And some of these regions were, interestingly, again, implicated in social cognition. And this was a really interesting result, we thought, because everybody, again, is getting the exact same linguistic input, and yet we're seeing sort of more synchrony. These regions are showing a more stereotyped response in the high paranoia group versus the low paranoia group. And I should also mention that we had several controls to try to rule out general attention effects here. So it was not the case that the high paranoia people were simply paying better attention based on their performance on the memory questionnaires. There were no differences in age or sex or other demographic variables between these 2 groups. So we think that this is sort of an intrinsic difference in terms of people's baseline paranoia that they bring to the table when they hear the stimulus. That factor is potentially driving differentially stereotyped responses in these brain regions. And of course, the nice thing about naturalistic stimuli is that we can then go back to the stimulus itself and ask whether certain events in the story are driving, or see if we can pinpoint specific events in the story that are driving activity in these regions. I'm going to skip over this slide for reasons of time. Right, so because we had created the stimulus, we had inserted events that we intended to be a bit more suspicious or ambiguous. So what we did here is we had a group of independent raters go through and mark moments where the main character in the story was experiencing an ambiguous social interaction or was explicitly mentalizing about the beliefs or intentions of other characters. And so we were able to treat this as a regressor, basically in a more standard GLM analysis. And our hypothesis was that some of the increased synchrony that we were seeing in the high paranoia participants was driven specifically by evoked responses to these events. And so we did this in a hypothesis-driven way in 4 different ROIs. We had our 2 main ROIs of interest. So these were the right medial PFC and the left temporal pole, which, if you'll recall from the previous slide, these regions were more synchronized in the high paranoia group than the low paranoia group. And we hypothesized that these regions would be more reactive to these mentalizing events. We had a positive control region in the left PSTS, which, if you'll recall from the group ISC map, this was a region that was highly synchronized in everybody, but didn't show a group difference. So we hypothesized that this region should show evoked activity to these events in everybody, but not necessarily more so in the high group. We also had a negative control here in left Heschel's gyrus, so just primary auditory cortex. So this region should not be differentially sensitive to these events in either group. And essentially, that's exactly what we saw. So in the 2 ROIs that were most associated with social cognition that showed this group ISC difference, the left temporal pole and the right medial PFC, we saw a stronger response to these mentalizing events in the high paranoia group. In our positive control region, we saw a strong response to these events in both groups, but no group difference. And finally, in the negative control, in left Heschel's gyrus, we did not see preferential activity to these events in either group. And so this was a really kind of neat confirmation that we can not only identify where group differences are using naturalistic stimuli, but we can also identify why. So in other words, we can go back to the content of the stimulus itself in a hypothesis-driven way and actually see which moments in the story are driving more activity in one group than another. I'm going to skip this for reasons of time, but we also had some interesting results from a naturalistic, from a free recall task that people completed outside the scanner. And we were able to identify signatures of trait paranoia in how people recalled the stimulus after scanning as well. So to conclude this second section, can we design a bespoke naturalistic stimulus to draw out variation of interest? In sort of this proof of concept study, we were able to use an ambiguous social narrative to evoke differences along a paranoia axis. And that brings me to the third section of the talk, which is this question of how should we model individual variability during naturalistic simulation? And so this was this sort of grew out of some statistical and implementation questions that I was grappling with during the paranoia study, to really understand sort of what is the nature of individual variability and how we model that really affects what we see. So to get into this part of the talk, this is sort of what I'm calling idiosynchrony here. So this is the general movement from using naturalistic stimuli to study shared responses to using them to study individual differences, which, if it's not already clear, I think holds a lot of promise. But I think that there's sort of a lot of both theoretical and implementational questions that we have to grapple with as we do this. And so Sam Nastasi and co-authors had a nice paper a couple of years ago kind of outlining this really powerful technique of ISC. And one challenge with ISC is trying to adapt it to the individual difference context. And I'll explain what I mean by that. So in this formulation that they put forward, essentially we can think about the response at a given voxel during naturalistic stimulus as a mixture of three different components. So x of t is the response of a voxel, and it's composed of this shared stimulus evoked response, c of t. So this is essentially what we can pick up on using ISC. This is the component that's shared in everybody. But then there are these other two components that are idiosyncratic. So there's an idiosyncratic response that's evoked by the stimulus. So this is something in an individual's brain, which is neural activity that is being driven by the stimulus, but is just unique to that person. It's not shared at the group level. And finally, there's this third kind of error term, which is stimulus unrelated activity. So this could either be neural activity that's just mind wandering, not associated with the stimulus. It could be physiological noise, basically anything that's specific to a subject and not driven by the stimulus. And so in this typical formulation, it's very difficult or actually impossible to tease out what is the idiosyncratic stimulus evoked response from the stimulus unrelated activity. So you could try to tease this out using repeat presentations of the same stimulus to the same subjects. And so you could kind of try to get at the idiosyncratic response by looking at things that are reliable within the same subject across repeat presentations, even if they're not reliable across subjects. But as we know, experiencing the same naturalistic stimulus a second time could also change people's reactions and interpretations to it. So another way that we could try to separate this id term from the error term is to try to anchor it to something else that we know about subjects. So we could try to anchor it to people's behavior score. And in this sort of general idea, you can kind of think of this as there's sort of a stereotyped response associated with a certain behavioral trait. And the degree to which a subject expresses that response is modulated by their behavior score. So kind of analogous to the paranoia study, there was this stereotyped response associated with high paranoia people that was sort of expressed more in the high trait paranoia people rather than the low. And so to make this a bit more concrete, one way that we can get at this, again, sort of reduce this problem of relating behavior where we have one measure per subject to ISC, where we have one measure per subject pair, is to treat this as a representational similarity analysis, or an RSA. In this case, we and others have called it intersubject RSA. And the idea here would be that for a given brain region, during a naturalistic stimulus, we can calculate a brain similarity matrix. Basically, you can think of this as an ISC matrix. So how synchronized is each pair of subjects in this given brain region? And we can relate this to a behavioral similarity matrix. And this then becomes a pretty straightforward representational similarity analysis. So if the upper triangles of these two matrices are correlated, we could infer that people's behavior score is somehow modulating their brain response in this region to the stimulus. So this seems nice and straightforward. But the question is, how do we calculate behavioral similarity? So in the case of certain behaviors, we may just have sort of one score per subject. So for paranoia, we had one summary metric. We might also be dealing with something like age. So we have a single scalar number per subject. How do we get a similarity matrix out of that? What does it mean for one subject to be similar to another subject? And the first thing that might occur to us would be to just use Euclidean distance or just subtract them. So a subject with a score of 20 and a subject with a score of 25, the distance between them would be 5. And indeed, this is how I think the vast majority of RSA would work, is that you're using this Euclidean distance here. And so this is simulated data. But essentially, by doing this, you're baking in a pretty strong assumption into your model. And that assumption is that subjects that score similarly to one another are also going to be similar in their brain response, regardless of whether they're generally high or generally low scorers. And so in this simulated matrix here, you can think about this as the subjects are ordered from low to high on this sort of arbitrary behavior score that we're talking about. And you can see that similarity is strongest along the diagonal. So people are most similar to their immediate neighbors on this scale, regardless of whether they're at the low end or the high end. And if we look at a two-dimensional embedding of that similarity matrix, we see this sort of linear shape here. And that's one plausible structure for this representational similarity. However, that's not the only plausible structure. In fact, it's not the structure that we saw with the paranoia data, for example. So in the paranoia data, we saw that there was a strong stereotyped response at the high end of the spectrum, but that at the low end of the spectrum, there was not a corresponding stereotyped response. So the low scores were just less similar overall to one another and to the high scores. And so if we think that that type of relationship might be at play, we would want to model behavioral similarity, not with the difference, but rather with something like the mean. So this is basically saying that subjects at the high end, so if I have a pair of participants and they're both high scoring, then the mean of their scores is going to be high. And they're going to have a highly similar response. Whereas if I have two low scores and I take their mean, that mean is going to be lower, and that's going to predict less synchrony. And you can also flip this around. This is for illustration purposes. So it could be the case that low scores are more similar and high scores are more variable, et cetera. But the point is that if we look at a two-dimensional embedding of this matrix, it predicts a really different structure. So here we're seeing, again, kind of echoing the empirical results that we saw in Paranoia, we're seeing that people with high scores are kind of clustered together here in the middle, whereas people with low scores are less similar to those high scores as well as to one another. So it's actually just an increase in variability. It's kind of a spreading out into the space. And there's a couple of other ways that we could model an overall similar structure. So for example, here, this is sort of a combination between these two models where we are still predicting a strong cluster of high scores, and then we see that low scores are spreading out, not necessarily anywhere, they're still spreading in a consistent direction, but there's also just much more variability among that low scoring group. And so these three structures might actually be more plausible for a lot of the behaviors that we're thinking about. But the point is that if you model them in the traditional way with just Euclidean distance, you're not going to be picking up on some of these effects that might be present in the data. And so in sort of thinking about these models and working out these models, we started calling this model the nearest neighbors model, because essentially this model is just predicting that you're most similar to your neighbors on the scale, regardless of where you fall in an absolute sense. And we started calling these models the Anna Karenina models, after the famous opening line of that novel, which I think goes, all happy families are alike, all unhappy families are unhappy in their own way. So the analog here would be kind of all high scores are alike, all low scores are low scoring in their own way. And so either of these models is plausible, and one might be more plausible than the other, depending on the behavior that you're interested in. But if you don't think about the way that you're modeling your data, you're not necessarily going to pick up on these effects if they're there. And so just to prove that these models really do matter, just a quick demo of IS-RSA on, again, turning back to the HCP 17 movie watching data. Basically, what we're doing here is we're using different models that make these different predictions to see if we can pick up on intersubject similarity during movie watching that's related to different behavior scores. So the first one I'll show you is just the working memory. So trait working memory ability. Do subjects with higher working memory ability look more similar to one another during movie watching in the brain? And we can model this again in different ways. So if we model this with the nearest neighbor model, we're basically predicting that there's a stereotyped response among low working memory individuals that's different from the stereotyped response among high working memory individuals. And we don't actually see that that model is borne out. So we ran this model at different regions in a whole brain atlas. We're using a replication and discovery, or a discovery and replication cohort here. So if this model were doing a good job, we should see a strong positive correlation indicating replicability across the two cohorts. We don't actually see much of that. We only actually see that two regions highlighted in gray are showing significant representational similarity using the nearest neighbors model. Whereas if we examine this using the Anna Karenina model, so this model is predicting that high scorers have a stereotyped response during movie watching. High working memory individuals have a stereotyped response, whereas low working memory individuals are just more variable. This model seems to be picking up on many more differences. So there's a much stronger correlation between discovery and the replication. The nodes highlighted in black withstands multiple comparisons correction. And so we're seeing that working memory in many ways seems to be an Anna Karenina type trait. So people with high working memory kind of look similar to one another, whereas people with low working memory just look more variable. So less similar to one another and less similar to the high scorers. We also tested this with personality. Interestingly, when we looked at individual personality traits, so this was the big five, which was also available in the HCP data, we didn't actually see much traction with either model. So we didn't necessarily see that people high on agreeableness had similar brain responses during movie watching. We didn't even necessarily see that people similar on agreeableness, as predicted by the nearest neighbor model, had higher synchrony during movie watching. So none of these models really did that well in capturing brain behavior relationships in this case. However, when we looked at personality in terms of item-wise scores, so now we're actually, instead of taking the summary scores for each of the 5 traits, we're taking just the individual items themselves in terms of how people filled out the personality questionnaires and just correlating the vectors of those items across subjects to get, this is our behavioral similarity matrix and essentially a personality fingerprint, so to speak. So how subjects tend to fill out the personality questionnaire. When we test that in our model, we do see a strong relationship with brain activity during movie watching. So we do actually see a decent correlation between the discovery and the replication sample, and we see some regions that show significant representational similarity. And what that means here is that subjects with similar personalities look more similar in terms of brain activity during movie watching. We can't necessarily say exactly why, so we can't necessarily say that this is being driven by a certain trait or a set of traits, but we are seeing some relationship between brain and behavior similarity in terms of personality. So to wrap up this last section, how should we model individual variability during naturalistic stimulation? I think we're seeing that intersubject RSA is a promising framework, but to think about our distance model and how which assumptions we're kind of baking in when we run these types of analyses. One last interesting question that I think is just sort of an interesting direction to think about, and I'd love to have a discussion on this potentially during the panel portion of this workshop with my fellow speakers. One thing that I think about a lot, just to sort of leave this off with a question, is how much ISC is optimal, right? So how much, if we care about individual differences and we're trying to use naturalistic stimuli to study individual differences, how much do we want people to look similar? So we want some degree of similarity, right? Because that's the mark that a stimulus is sort of at least engaging people to some degree. But is it the case that if we have a stimulus that's too strong, we could actually saturate people? So if we want to study trait anxiety, for example, and we show people a horror movie, maybe people that even aren't naturally anxious might look really anxious or might become really anxious during a really strong stimulus like that. Or maybe even with a stimulus of that strength, the people that are naturally more anxious or scared would still have sort of a differential response over and above that. So, this is an empirical question. I think it's just really interesting to try to think about the relationship between how much synchrony a stimulus evokes and the sensitivity of that stimulus to individual differences. And sort of trying to, this is again simulated data, but just trying to think about what this curve might look like. It could be the case that a stimulus that evokes only, a fairly low degree of synchrony is the most sensitive to individual differences. Or, you could want sort of this sort of Goldilocks stimulus, like somewhere in the middle where there is, a decent amount of synchrony, but there's still room for pairs of individuals to vary in how much synchrony. Or, maybe a strong stimulus really is best. And maybe even stimuli that really strongly sync up different brains are still sensitive to individual differences over and above that. So I don't have the answer to this now. I just think it's sort of an interesting thing to think about and potentially a discussion point. Next steps in my lab that we're really interested in doing, we're currently analyzing a data set with 4 different indie film clips. 3 of them are social in nature. One of them is a mechanical film that's just meant to be a sort of control. And we're looking at, we have individuals that have on a range of symptoms of mood disorders, and we're interested in seeing which film best pulls out differences related to those symptoms. And we're also really interested in figuring out where along the processing hierarchy we see these differences. So one prediction might be that at the low end of the feature hierarchy, so really basic components of these stimuli, like just loudness, frequency, luminance, and flow. So audiovisual properties, we might see less individual variation. And then we can sort of move up the processing hierarchy to think about semantic categories like faces and places. And then at the highest level of the hierarchy, we have things like language and emotional tone. And so one prediction might be that, again, simulated data, but if we think about each individual gray line as one participant and the black line as the mean, we might predict that most individuals look similar in their response to these low-level stimuli, whereas the further up the hierarchy we get, the more variability we might see. And so trying to parameterize that and figure out where we start to see these individual differences diverge in terms of this processing hierarchy is one thing that we're really interested in exploring. So with that, I'd like to thank all of the collaborators that were involved in all of the work that I presented here today at my former institutions, NIMH and Yale. I'd also like to thank the members of my lab at Dartmouth, the Functional Imaging and Naturalistic Neuroscience Lab. I'd like to thank my funding sources and leave you with this sort of last image of the dangers of averaging data across subjects. So thanks again for this invitation, and I'm really looking forward to the discussion.